Hello, good morning, everybody. I hope you're all happy, healthy, and well on this beautiful sunny San Diego morning. Welcome to week 7 of BIMM-143. Thank you for joining me, either live here or recorded, as we'll see in a moment. I'm just going to make sure I don't get any feedback here by turning off my screen. And so this week we talked about, we began our introduction to machine learning. We mentioned what unsupervised learning and where we focused, but also supervised and so-called reinforcement learning that we got delved into in our recorded videos. Here I am down here in this little postage stamp, and then we got into the two kind of main areas that we talked about for this week, or that we focused on learning for this week. That's clustering, and again we went into two major methods in this clustering realm. That's K-means clustering as well as hierarchical clustering, and we'll have a little review of those in a moment. And then the main one, really one of the most useful things we've learned to date in this class, that's dimensionality reduction, how to really find features in our data and structure in our data that might be indicative of the patterns or the biology or the mechanism that we're actually investigating with these different data sets. And there we focused on this method called principal component analysis. And that's really what we're going to focus on in the lab session for this week. We're going to apply these principal component methods to a number of different data sets, and then also supplement them by applying clustering within or built on the principal component analysis results. And we'll see how that kind of works in a type of pipeline for data analysis. Okay, so let's get started with a brief review here of clustering methods. So in those videos for this week, we talked about two main functions and two main approaches. The first was K-means, and the function to do K-means in base R is called K-means. Funny enough, right? Who would have thought it? So here in this slide, and we had this slide in our video too, we make up some data just to get us started, to whet our appetite, and before we move on to more real-world data sets, including some real cancer cell data in a moment, as you'll see. But here what we're doing is, in the first line of code up above, we're generating a little vector called temp, TMP, because I'm not very inventive, right? And that is taking input from this rnorm function. Rnorm, I'm asking for 30 points, centered at -3. That's the first part there of the first line. And then there's a second rnorm call, which goes and gets another 30 points centered at +3. And then the second line, the X here, this thing that I'm going to plot eventually and actually do some clustering on, it puts them together. One the straight way around and the one reversed. Maybe what we'll do here is we'll actually just go and take this to RStudio and see what the effect of plotting X is and dig into how X works before we move on to answering these questions that are in the white box here. That's how do we do clustering on this data set? How do we interpret the results that K-means gives back to us? And then we'll move on to hierarchical clustering and do the same thing in a moment. Okay, so let me pop over to RStudio here for a second. So here I am in a fresh RStudio window, and we can open a new, of course, same as always, we'll open a new project for today's or for this week's work. So I'm going to do File, New Project, and we're in week 7, so I'm going to open a new directory, new project, and I'm going to put it where I'm storing all my class material as always. You know, I sound a bit like a broken record here, but it's really important that you remain organized. You'll see in 2 weeks' time, when we go to put lots of these things together into a nice portfolio, that if you aren't organized, if you don't know where all your stuff is, it's going to be a little bit more difficult to make that kind of portfolio. So we're going to keep ourselves organized. We're going to work in the same directory as always, but this time create a new project called, I'm going to call it week 07, and it's in this location where I'm working off my desktop for this class. So I'm going to click Create Project. RStudio will flash and launch R running in that location, in that folder on my computer. I can see that because I can see this up at the top of this first panel window here, I can see this thing here. If you can see where my mouse is, right up here, this is the location that I specified with the week 07. You'll also see up in this other corner in the very top, right above my head up here, at the very topmost of the window where the mouse is, you'll see it says the name of your project. So you know you've successfully done it. We're in this new project. I'm also going to open a new R Markdown document to store my work for today. So I'm going to do that, File, New File, R Markdown. I'm going to call it week 7, week 07. Actually, this is the title, so I'll call it Machine Learning 1, number 1. This will be the title of the document. I'll just leave it as HTML for now, and we'll change it to PDF a little bit later. So this opens up a new document with this boilerplate code in it. It's got the title that I just typed in that box here. We could change it if we wish. We could change the output format here by editing these first 6 lines here in this YAML header document. All the rest that's below this setting chunk from line 11 onward, I'm just going to delete because I don't need it. So I'm going to delete all that. I'm going to save my document here, my R Markdown document, because it's called Entitled 1 and it's in this blue font. I know it's not saved. So I'm going to save it here, and this is where I'll actually give it a file name. I'm going to do week 07.rmd. And we should see that file now appears in this window right behind me here. It says, where is it? Right there. Week 07.rmd has just appeared in the Files panel. So now I can start playing. So here I'm going to do Kmeans clustering example. So we're kind of getting started with Kmeans. Right. Now the little X that we made up, I could type it out things. So I think we did temp, and this was you could copy it from the slide if you had it, but I'm just going to type it out again. So we make a vector. C is the function to make a vector. So I have my parenthesis in here. I'm going to make this vector, and the first one is I'm going to do R norm. So I'm going to, this function here, what it'll do is it'll give back points. Let me make the font a little bigger, perhaps, so you can see it more clearly here. So I'm going to ask for 30 points, please. So that's the first parameter. And then by default, they'll be centered on 0. I'm going to say don't center these points on 0. I'm going to say center them on -3, please. And then I'm going to ask for another 30 points from a different center position. So I'm going to 30 and center them at +3, please. So if I run this code, I actually have made a little boo-boo here. I haven't put it in a code chunk, so it ain't going to work, right? If I go to knit this document, it's not going to run any code, right? This was a thing that we had discussed in Piazza the last day in the last session. My code doesn't run when I knit my document. There's no results, and that's because it isn't inside a code chunk. Art just thinks that this is good regular old text, right? Because that's what I've written it as. So I'm going to close this down and do it properly here. I'm going to actually go and take this code chunk. I'll give myself some room here with a few return carriages so this thing appears towards the top of our screen. And this thing should be inside a code block. We can insert the code block in a number of ways, as we saw before. We can do the button that's up the top that says insert that I've just clicked on, and this is our code, so I could do that, and this will do it too. I could also use the shortcut key, right? Command Alt I or Control Alt I, and that does the same thing. Or I could type it out, the little 3 backticks, square brackets, or sorry, curly brackets, the kind of mustache brackets, or whatever you want to call them, and the name of the language we're using R in this case, and we put that inside. And make sure you close them, right? You need the 3 to close. So when I do this, this is going to print out a vector with all these numbers in it, right? These random numbers. If I knit this document, I'll actually have output in it, right? I'll see here it's done something. It's in this code chunk and there's the results down below. Okay, so that's the idea. We want to keep the code in code chunks and the text out of code chunks. So let me close that. What we actually did was we, in the slide, we made another vector called x. And here we're just going to bind these together, cbind, so bind them by column. And we're going to call one x equals temp, and then y equals the reverse of temp, something along those sort of lines. So let's have a look at what x looks like. So we saw here below that in the console when it printed out, this was just a vector of 60 points, 60 elements. There's a lot of values centered around -3, and the values centered around +3. What we're doing here with the cbind function, as we'll see in a moment, is we're putting this copy together. So here's all these points in the first column, and here's all these points in the second column. So we've got two columns and 60 rows in this thing. We could check that here by saying the dimensions of x. So we're just making up some toy data to play with. Okay, so it's 60. The first 60 here says 60 rows. The 2 says 2 columns. And I'm just going to plot it. Plot. I'll use the base R function, plot. You know, we don't need to be too fancy here with ggplot or anything else. We're just going to go and have a quick look. And when I plot that, here I see there's 2 clearly separated constellations, if you will, of points. Okay, so what we're seeing here is this effect of taking the x vector and plotting it, right? These 2 constellations. So we're going to go, and the main purpose here is to do k-means clustering on these points and see how this thing works. So the function to do clustering here is called, I'll write some notes for myself about what I'm doing here first, as you should. Let me actually fix the audio for a second. I'm going to mute that audio. There we go. So back here, I'm going to write some notes for myself. Here, make up some data to cluster where we know the answer, know what the answer should look like. Like your eyes could tell you with that plot that we produced here that there should be 2 groupings. That's what clustering is trying to find for us. It's trying to reveal these groupings, these clusters that are distinguishable from each other. They're more alike within a cluster than they are between clusters, and there's 2 of them here, right? Our eyes can see these 2 groups of points. So how does k-means go about this? And of course, in our online video, we talked about how k-means works. We walked through the algorithm. The function itself is called k-means, and we're going to call it. We give it the input that we want to cluster. I'm going to give it x, because that's the thing that we plotted up here. And k-means also takes, if we pull up the help for it, or if we look back at those videos, it takes the k, right? In this function, it's called centers. How many groups do you want? You have to tell k-means at the start how you want to cut up this data and split it up into groups. So we're going to do centers. I'm going to let RStudio help me type it correctly. It'll prompt me centers. I'm going to say 2, and I'm also going to set this optional third argument to run it many times, right? I'm going to run it 20 times, iterations, because you remember it has this random component to it. K-means, it goes and it starts and picks some of our points and says that's our initial cluster 1 representative, that's our initial cluster 2 representative, and then it goes through and assigns all points based on their closest distance to those points and comes up with an answer, and then looks at the sum of squares, the spread within that answer, and then does it again, picks another 2 points, and then does it again, does it again, does it again, and then goes back and looks at what are essentially the most compact clusters and tells you about that answer. So the more times you run it, the more opportunity you have to find a good clustering. So I'm going to need an object name to store my results in. I'm just going to be uninventive and call mine K. And then I'm just going to print out K. What is K? Here, so the line of code here runs the Kmeans function and then prints out the results. I'll show you. Let me turn the plot off so you can see both those lines, or all those lines together. Okay, so we made our little vector X with those two groupings in it. I run Kmeans. Let me squish this over a moment. It tells me Kmeans clustering with two clusters. We told it centers equals 2, so it's telling us 2 clusters. And it's telling me the sizes of the clusters it found is 30, 30. And that's what we expect, right? That's good, because we actually, when we made up our data, I asked for 30 points here and 30 points there. And so the cluster sizes should be 30, 30. So that's great. It tells me what the means are, the cluster means. So this would be the centroid, or the center point of those clusters in our XY plane here. And so it's telling me those are around +3, -3, and -3, +3. And that's, they're not exactly that because we're random numbers around that position. Your values will also be slightly different because you're pulling random numbers with that R-norm function, so we're going to have slightly different points, of course. But this is good, right? We've told it 30 points please at +3, 30 points please at -3, and it's telling us that that's exactly what it's found here. And it's also giving me this thing called a clustering vector. What this thing is, it goes through the input, all the rows there of that input, and remember there were 60 of them, so this vector will be of length 60. So that's for each row that we have, which cluster has it been assigned to? Is it cluster 1 or cluster 2? So it's assigning here the first, if we count up, here's 22, there's 21, first 21 rows are in cluster 2, then 22, 23, 24, all the way to 30 are in cluster 2, and then the next 30 up to the 60th point, from the 31st to the 60th, are in cluster 1. Okay, so we'll see why that's useful in a minute, but basically it's our answer. It's which cluster do our points belong in, this clustering vector, sometimes called a membership vector. And then it tells us this within cluster sum of squares. That's how it measures and keeps track of what's the best answer here. Remember when the algorithm works. And it tells me available components. So these are actually all the things that are available in this object K that we just created here, K. We could go and use $cluster to find this, $size to access individual components of this object. So let's try that. Actually, the questions were, if we were back on the slides, as we've run our clustering, if I pop back over here to the slide just for a second, the questions to remind ourselves were, how many points are in each cluster? Well, the print method tells us it's 30, 30, but how would we actually get that from the result object and incorporate it in our report, for example? What component of our clustering result details the membership vector and the centers? So let's see how we would actually access those things from this Kmeans result. So I'm back here in RStudio. I'm going to say type some text to describe what we're doing, some questions. How do we get to our results, right? So how do we get to our results? Specifically, question 1 would be, how do we get to the cluster size? Cluster size. And so we saw here that there was a component called size. And we could actually so K was the object that we got from running Kmeans. I'm going to do K, and I'm actually going to look for the attributes. This function attributes, if we pass it, this thing, it'll tell us what's available for us to go and inspect. So when I run K look at the attributes for it. It tells me there are these named things. It's a list object actually, and it tells me I can go and access cluster, I can go access centers. There's one called size. That's probably what I'm after, right? So let's try it. We could look at the help page, of course, of Kmeans, and that would tell us too. Actually, that's a good idea. Let's go and look at the help of Kmeans. So I'm just going to put my mouse over this function. I'm going to push the F1 key on my keyboard, or to pull up help, you could do the question mark, the name of the function within your console. And RStudio makes it kind of easy with pushing the F1 key, function 1. And over here in the help panel, it tells me here's Kmeans, here's all the different approaches, the different arguments like centers that we set, the end start that we set, different methods like complete or single or average, linkage that we talked about in those videos, and then quite a bit about the algorithm and how it works. But down here, when it says value, that means it tells us, the values that are returned, what's actually available in our result object that we can get to. And what we're looking for, our first question over here was cluster size. So we could access $cluster, $centers, total sum of squares, or tot SS, and here's one called size, the number of points in each cluster. Okay, it's kind of obvious that that's what it was. I'm torturing you here a little bit with this, but I'm going to do $size. And if I go and run that, if I click the little play button, it tells me 30 points in the first cluster, 30 points in the second cluster. So how about the membership vector? Actually, our main results here. Which cluster does each of my data observations, each of my rows actually belong into, right? So for this, I'm going to use $, and I could, so RStudio is telling me these are all the things that are available, cluster, tot SS. I can scroll down through them. I'm going to pick cluster, because I read it over there in the help page, and it gives me that vector back. Remember the 30 points that are in 2 and the 30 points that are in 1. We could double check how many points are in each cluster. And we could do that with the table function. Just go please and tabulate up what's inside that $cluster for me. That'll give me a count of all the 2s, how many 2s there are, and how many 1s there are in that vector that we just printed out above. So if I do $cluster, here it'll tell me 30 points in 1, 30 points in 2. So everything looks good. It's meeting our expectations here, and we're good to maybe go on and look at the final 2 questions. The next question was, I think, about the cluster centers. Okay, we get the idea. Cluster center points. This is going to be in, you guessed it, centers. Okay, so this is going to give me the actual coordinates of the center points. There's 2 clusters, so here's the first center point of that group, and here's the second center point. So the last question was more, okay, that's still a little bit abstract. Can we just plot the results? Can we visualize them and see how things look? Okay, so make a plot. Let me put a capital for our Kmeans results, colored by, so here specifically we want to color colored by cluster, with cluster centers also shown. Sorry about my keyboard's a little loud. So let's try and do that. So here, remember that we could just, before we could just, as we did up above, we could just plot X. That was the data, the input to Kmeans, and that produced this rather unexciting plot with the 2 groupings, or the 2 sets of points, but no indication of which cluster is which. I can actually go and pass it here. So the cluster membership vector. So this cluster membership vector we printed out here. It's called $cluster. This is this vectors of 2s and 1s. And this 60, it's of length 60 as we saw, and has 31s and 32s in it. So I can actually pass that to the color argument of plot. Call equals $cluster. And what that's going to do, it's going to say color the first point. In our case it was 22222. It'll pick the second color from the color palette, and whenever there's a 1, it'll pick the first color from the color palette. So if I do that and run this, I'm going to get, first color is going to be black, second color is going to be red. If I'm going to do this, it's going to get red and black points. Okay, so there's our 2 clusters. So that might be when we used base plot before and we've used ggplot2, of course we often use named colors like red in quotes or black in quotes or blue in quotes, these sort of things. But we can also just color by number. Let's make sure we understand this. So let me delete this for a second. We could say, go please color everything the second color in the color palette, and every point is going to be colored the second color, which corresponds to red. Black is the default, the first color, and you would see the third color here is green, and it goes on and on up the color palette. But when I pass a vector of 2s and 1s like I did, so $cluster, it of course has that vector that we saw up here. The first point's going to be colored red, 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 red. There's going to be 30 red points, and then there's going to be another 30 black points. That's the idea. So we're using this ability to color by number as a way to quickly show that feature here. The question also asked, can you show the cluster centers? So with base plot, I can use points. So points will just add points to an existing plot here, and what I need to pass in are the coordinates. So that's $centers in this case. And if I just do this, what's going to happen is there's just going to be 2 black plots. I don't even know which point it is here in the second black group. So I'm going to color it differently. So I'm going to say call equals, let's color it blue, for example, here. So again, it's not too easy to see. There are, probably on the screen resolution here, it's not great. So let me change the plotting character. So PCH, let me do a square, 15. And if you didn't know these parameters, and I don't expect you to, we covered them back in class 4, you can always look them up. Google will help you. How to use a square plotting character, BSR plots, and it'll tell you how to do it. So here's a little blue square. I'm going to actually make it a little bigger so we can see it. So I'm going to change the character expansion, CX. Let me do it 2, for example. And here we've got 2 big blue squares. That's the cluster centroids from which this sum of squares distance is measured, of course, right? Great. So that's our Kmeans results for now. The purpose there was really to see how to get to the results, how to get to that cluster vector, that $cluster, the membership vector. Let's go on to hierarchical clustering, and this is so next we're going to move on to, and I'll actually go back to the slide for a minute, but the idea is somewhat similar. It's a little less straightforward to use the function, although it's actually a lot more powerful clustering method because you do not have to tell it k beforehand. You don't have to tell it the answer beforehand like we did with Kmeans, right? But this is the Hierarchical function. And we're just going to, just for demonstration purposes, analyze the same little baby data set there, X, that we created above. So we don't have to type that in again. It's the same thing that we had from before. And we're going to use Hierarchical. But Hierarchical isn't as straightforward as Kmeans. It doesn't, underneath the surface, do the distance calculation for you of each point to the cluster center and work it all out. You actually have a lot more flexibility with Hierarchical. You can give it different distance metrics or measures of dissimilarity, like sequence identity or structural divergence or whatever, RMSD or whatever kind of measures that you're interested in depending on your data. and then pass it to hierarchical clustering. Let's see that. Let's go back to RStudio and quit our blabbering and actually just do the thing. So here I'm going to, once I find my pointer, here I am. So I'm going to here do the same thing here. I'm going to do hclust, and I'm going to actually pull up the help page for it. So I'm going to push function F1, or you can do question mark hclust, and this is hierarchical clustering. I should put a title in here. So we'll cluster the same data as before, but using hierarchical clustering. And we'll see that hierarchical clustering over here in the help page, it takes an input called D, and there's different methods here. It takes a dissimilar data structure as produced by the function dist. So we need to give it a distance matrix as input is the key here. So we're going to give it dist of x over here, and then I'll leave it as the default method, and I'm going to save my results as hc here. You know, you pick whatever name floats your boat. I'm still a little uninventive, and I'm going to, just like we did with Kmeans, I'm going to print out what hc looks like. We'll see that it's not that exciting for hierarchical clustering. It basically just tells you what you did, right? Here it's telling me, you know, the default method, clustering method, was complete linkage. The distance matrix that I'm giving in, the default for dist is Euclidean distance. So that's, you know, our regular Cartesian kind of distances. And then the number of input things that we give, there were 60 rows, remember, so it's not really very helpful. The print method just tells me, you know, what I already knew, what I give as input. But where hierarchical clustering really comes into its own, of course, is I didn't have to tell it centers. I didn't have to tell it, you know, give me 2 clusters, please. And then Kmeans will go off and give you 2 centers. Even if you give it, you know, give me 10 clusters, please, Kmeans, it'll do it for you, right? It loves you, and it'll faithfully go and try and produce the best 10 groups that it can. That'll maybe meaningless, as it would have been in that previous example, but it would have done it. Here, hierarchical clustering won't impose a structure on your data like Kmeans will. You know, if you ask it for 10 clusters, it'll give you 10 clusters, whether that's sensible or not. But what hierarchical clustering will do is reveal some structure in your data. And I'm going to do that. I'm going to actually plot hc. So this isn't going to produce a scatter plot. This is going to take advantage of the object-oriented nature of base R, the class that the hc object R knows that it came from a hierarchical clustering method. So when I plot it, it's going to produce this tree-like structure called a dendrogram. So I'm going to plot that. And here it's a bit squished in my window. So I'm going to open it up a little bit here in a larger window. And then we'll have a look here, and we'll see if you notice anything about this clustering. Okay, so we talked in our videos here about how this is made, how the hierarchical clustering process goes, and in this case from the bottom up, and takes the 2 most similar points and puts them together and draws the little goalposts right at that particular distance, that height here, and then goes and does the next ones and the next ones. So you get these increasingly tall goalposts. And that's what's happened here. But do you notice anything about this? Where's the biggest goalpost? What's the easiest goal to score here? Where would the goalkeeper have, if there was one, have the most difficulty? It would be this big one, right? It would be, this is the largest upright bar here, right? So this is telling me that there are 2 main groupings. There's this section over here, and then there's the second one, groupings of points right over here, okay? So it's revealing something about the partitioning of our data, about the structure in our data, this tree. And that's why we use them, right, to look at this kind of structure. And then the labels here, you know, if we were working on our, you know, genes or proteins or whatever, and they were row names, it would actually label them with the row names here. I didn't set up X with any row names, so it's just going to call the first row, the first point 1, the second one 2, the 10th one 10, the 50th one 50, the 60th one 60. So the labels down there are just which row it is in our data point. Do you notice anything about the partition of these points? Like here in this first grouping over here, for example, do you notice that the numbers, are they big numbers? Are they above 30? Or are they small numbers? Are they below 30? Well, if you look at them, you know, there's 8 and 12 and 13 and 5 and 2. There's no number, there's 30, but there's no 31. There's no 32, 33. This is the first 30 points. That's that first grouping of data points that we had. And then over here, in that second main group, second main cluster, if we cut this tree, as we'll do in a moment, we have the points 31 to 60, right? So this is what hierarchical clustering is doing and showing us this structure in the data. So let's actually, I'll close this down, this big one, but what we're going to do here is we're actually going to draw a little line across, just for visualization purposes. I'm going to add a line, and I'm going to add it at ab line height equals 6. So it's going to draw a line at that coordinate across 6 here, where those goalposts are, and I'm going to color it just to be more visual. I'm going to do red, and yeah, that'll do for now. So let me draw this line across the tree. Maybe I'll do it at 8, just so it's a bit higher up. And so you see this red line now that's drawn across at the 8 position. It's like what I want to do here when I view this thing and start to make sense of it is I want to take a saw and cut my tree along this red line. And what I'm going to get is 2 big branches. I'm going to get that dangling big collection of leaves on that first branch, those points that are 1 to 30, and then I'm going to get the dangling leaves that are 31 to 60 in that second one. And that's the job of the CutTree function. So CutTree, what it's going to do, it won't draw a line for us, but it'll cut it. Let me do it in a new code chunk here. I'll actually close this down so we can see the code, what's going on. I'm going to do CutTree, and I'm going to tell it, we have to tell it the tree that we want to cut, and that's HC, the dendrogram that we want to cut. And we can tell it the height that we want to cut out. We could say height equals 8, just like the line we drew. And what this is going to return for us is the answer, the membership vector. Remember the same thing we got from Kmeans? We got it much more easily from Kmeans, of course, right? Because it was just there in the result object. But here we have to tell hclust, where do we want to cut this thing? Where do we want to draw this line? We can also tell CutTree. Sometimes easier, especially when you're dealing with much larger things. We can also tell CutTree here. Oh, sorry, need to put it inside a code chunk. We can also cut. Just say, can you please go cut the tree to give me 2 big groups? So it'll go and cut it at the height automatically for us. So I'm going to go CutTree, same thing, HC, and in this case the argument is K, give me K groups, please, give me 2 groups. So it's going to cut the tree into 2 groups and spit back that membership vector. And it's the same, obviously, as the one before, because it's equivalent. Whether you cut it 8 or you ask for 2 groups, it's going to give you the same answer. This is our result from Kmeans. And I'm going to go and plot that up. And actually, I've been talking for a long time. I apologize. Let me see if there's any questions after I make this little plot, and we'll go on and we'll talk about this a little bit more based on those questions, and then we'll take a break and we'll go into principal component analysis. Actually, the more interesting part of this thing. So let me first plot this up, and we'll then go and look at question responses if there are any. So I'm going to do plot. Remember, it was x, and this time, where do I get my color vector from? Well, I'm going to go and get it here from the answer to CutTree. So if I had saved this, for example, I could save it into a little vector called groups, for example. Oops. And if I run that, then I would have a groups vector that I could add here, and I would color up my results by the hierarchical clustering vector. Okay, so that's a lot. Let me turn off this plot for a second. I'm going to show this code or leave this code up where we do our hierarchical clustering. Cluster the same data as before. Hclust function requires a distance matrix as input. So it's different than Kmeans. It's a little bit more complicated to use. We get that from the dist function. And then we cut the tree to find the membership vectors. So I'm writing notes for myself, right, for my future self here, so I know what the hell's going on when I go back to look at this. If we're doing a project, for example, it would be useful to look at this file in the future to find out how to run these methods. The cluster membership. To find the cluster membership vector, I.e our main results. We use the CutTree. I'm putting the functions, or the R code, in these little backticks, and you see they're being colored distinctly. So when it renders, I know that these are code, right? And I can use CutTree in either two ways. I can give it a height, or I can give it a K for groups. You could write a note about that. Let me render this, and as that's rendering, we'll go and look at questions. I see, let me check the chat window. So Ali says there's an error message, couldn't find function CutTree, and it's CutTree with only one T, so it's just spelling. 99.999% of the time errors with CutTree is because I write two T's, because it's CutTree, but it's only one T, CutTree. It's hard for me to say, but it's CutTree, as you'll see in a second. So there's our Kmeans results. With our cluster centers. Here's the dendrogram, looks pretty, and our hierarchical clustering results. Let me pop over also and check here for a second on, I'll bring the window over here too, over to our website. I think Alina's answered this one. So when you, the poster here is asking, when I do my $K centers, I get slightly different answers than I got. And that's because, and everyone will get slightly different answers. If I run that code again, let me see it, let me show you actually. So here, I knitted the document. So that would, in essence, give me, it'll run the code again. See the centers that I get? So I have to actually show this. If I go back to RStudio. So here's my knitted document. So that, when you click knit, it goes and runs all your code and embeds it in the output here. So when I type $K centers and it runs the document, I get this answer, -3.00, whatever, right? I should round these things up. If I go and look on the console and look at this, it's different, right? The answer is slightly different because we're using that our norm function, the random function, when we made up x and temp that we built it from all the way back up here. This thing will return slightly different answers every time because it's drawing it randomly, right? That's why it's got the noise, all the points just aren't on top of each other. So it's kind of on purpose, right? So you're going to see slightly different answers. So let me go back and see if there's any other questions. Feel free to post along on Piazza. Or in the chat window here. Probably Piazza is easier for threaded questions. What is a distance matrix and why is it necessary for hierarchical clustering? Alina's done it. A distance matrix is what it is a pairwise. So we have 60 points in our input here at the minute. So it's going to go for all of those 60 points and say what's the distance between point 1 and point 2. Point 1 and point 3, point 1 and point 4, point 1 and point 5, all the way to 60, and then it's going to go on to the second point and say what's the distance from the second point to the third point. It's already done the first point, fourth, fifth, all the way down to the 60th point. And then it's go to the third one, what's the distance? So it's these pairwise distances in space. So it's just using little, Pythagoras' theorem, right, to find out that distance, that hypotenuse of that triangle in this case, and that's going to be the distance matrix. So this square matrix, it's square because it's n by n being, the number of data points, so 60, so it's 60 by 60 matrix. That's the thing that goes in the hierarchical clustering. That's the distances that then hierarchical clustering uses. And as I mentioned, we have to calculate this before we do hierarchical clustering, because hierarchical clustering doesn't do it for us. Kmeans did. It would actually go and calculate the distance matrix under the hood for us. We don't have to supply a distance matrix as input. It does it for you. Hierarchical clustering doesn't do that because it gives you the ability to to provide lots of different measures of dissimilarity. We just used Euclidean distance, but that comes from the standard dist matrix. But we could have used any other measure of dissimilarity, right? Like Hamming distance or sequence identity or sequence similarity or something else if we're looking at sequences, for example. We'll often do that kind of approach. Okay. So let's take a drink of water and a break, and then when we come back here in a second, we're going to delve into hierarchical clustering. Let me pull up the slide deck here, and we're going to move into, not hierarchical clustering, sorry, but principal component analysis. Okay, so I've got my breath back, at least temporarily. So principal component analysis, this is really, it's hard to overemphasize how useful this technique is, and actually the whole family of techniques that are embodied in this dimensional multivariate analysis approaches, TSNE, UMAP, and others that will may come across in this field. They're all kind of have the same motivation. They want to reveal hidden structure in big data sets, right? Data where you're measuring lots and lots of things, like we do all the bloody time in mathematics, right? We've got these confusing ass data sets with lots of things we're measuring, 20,000 genes or more over lots of different conditions and cell types or cell lines or drugs and non-drugs or patients and this and that, and we've got these huge data sets, and you can't just plot X versus Y like we just did, right? It's just impossible to see structure like that. We need some method to try and help us get to the essential essence of that data, right? What is it in there? What are the patterns? What are the groupings? What's going on? And this is where these multivariate analysis methods, of which principal component analysis is just one, but it's a very good one and a good place to start, is a very useful approach. So now, this can be, and I appreciate this, a little bit of a mind warp, right? It was for me when I learned these things first, a long time ago in a galaxy far, far away, right? So when I was young and more into these things, right, and engaged in talking about these things, when I learned this kind of stuff back in Ireland, back at that time, it was a different world, right? It was a different kind of class that I would learn this in. You would never find this in a biology class like this one. Where I got it was in kind of linear algebra classes, just like this tweet here from this person, David Robinson, saying, every linear algebra class, it's like we learned the math behind it, how to do it, how to do the diagonalization, the covariance matrix, etc., what it meant in the math, and that's fine, that's one way to understand it, but I didn't know how to think about it, right? That was the problem. I didn't have a sense for what it was actually doing or why you were doing it. So just like this person saying, the teacher here is coming back with a math answer, you can think of these eigenvectors, which are principal components, as an n-dimensional kernel subspace. It's like, no, right. That's not a great way to think about them for us, for biologists. We're motivated by analyzing our data and finding patterns in the data. So the way I'm trying to teach it is not with the math, not with the linear algebra, but really talk about it in a more pictorial, more practical, more hands-on way, where we just go and analyze different data sets and get a feel and a sense for what these methods can do for us. And then we can come back, as we will in a bit, and talk about the math and the basis of it. So what principal component analysis does is it's going to take all those things we measure about our genes or our proteins or our patients or whatever it is in our data set, and take them and project them onto these new variables, these new axes that we call principal components or eigenvectors, that are better than the original things we measure, in the sense that they'll show patterns to us in a much more clear way, right? The motivation is really to reduce the number of things we have to look at and the number of things that have to make our mind go in our data sets, but show it in a small number of dimensions. Dimensions just means, first dimension would be a number line, second dimension is a 2D plot, third dimension, 1, 2, and 3 is a 3D plot. But we're often dealing with hundreds and if not thousands of dimensions. You can't just plot that stuff, right? So what it's going to do is take all those dimensions and squish them down, but retain the essential essence of what's I sound like a shampoo commercial, right? But it really is. It's the essential features of that data we want to retain in these principal components that are going to help us figure out patterns and groupings and structure in our data, similar to the way clustering tries to do things. So what I've got here in this little plot on the slide beside me here is just two axes, right? Typically we have thousands of axes, but I'm just drawing two, and we've got points, these red points in it. And what principal component analysis does here is these PCs, or these eigenvectors, they follow this kind of line of best fit that lies closest to the data. So that first axis, the yellow one, that's the line of best fit, the best one that describes that data, right? So we can take that, and the second one describes the second most. The principal components, just like that math professor was saying in that tweet, you can think of them, because we have many dimensions, not like a line, but a surface, right? That's what he's saying. But really, let's just think of them as a line for now. They're a new axis, a new line that we can use to describe our data that lie closest to those observations, okay? So, and then what we can do, and see my fancy animation, we can throw away all those original things that we're measuring about the data for a moment, those original axes, get rid of them, and use these new, better axes, these principal components, to characterize our data, to plot it and analyze further in this basis set, rather than the original lots and lots and lots and lots of thousands of dimensions or whatever it is. We can go take a small number of these principal components that do a better job than the original ones at describing our data. So we'll do that. Now the first principal component, they're ordered, PC1, PC2, PC3, all the way down to the number of original dimensions you had in your data, in fact. But we typically confine ourselves to the first few principal components, because they're ordered by how well they do, how good they are at describing the original variance. So variance, let's just think about it like the spread in the data, like these red points, they're spread out most along this first line here, this first axis. The first principal component, this one, does the best out of all of the principal components at describing the spread. It describes as much as it can, and then the second principal component is going to describe the rest as well as it can, right? So so the first component here is describing that left-right kind of variation in our data, or variance in our data. And then we need a second component here to describe the kind of up-down variation here that we have that is not captured along when we project to that first principal component. And then there's a third one, etc., etc. So they're going to be less important the more you go down the principal components, the more you go up the number. PC1 is always more important than PC2, which is always more important than PC3. And we quantify that by the amount of variance, also known as the eigenvalue, the thing we get from that diagonalization, from the math part of it. Okay, so let's just hold this thought for a minute. There are these lines or surfaces that lie closest to our data, and the reason we do it is to give us a better, more hopefully coherent, from my little mind's view of our data set and the spread in the data. That's why we do it. So hold on to that thought for a minute. Let's go and analyze some data and see what they look like, and then we'll come back and we'll talk about how it actually works, how it works under the surface. Okay, the objectives here, of course, to reduce dimensionality. So dimensionality here, the terminology just means all those things we measure about our data, all those genes and all those different conditions. Please make it simpler, put it down into one figure I can look at, right, for example, to visualize that multi-dimensional data set. Then we can do analysis in that basis set, those new axes that do a better job than the original ones. We can do clustering in it, for example, as we'll do in a moment or two, rather than doing it in the original dimensions and getting confused again. We'll do it in the new basis sets. We can then identify clusters or groupings or structure in our data, and we can also identify weird things, right? If there's one point that doesn't sit anywhere near any of the other points, it's probably something we want to delve into. Why is that single point different? Is it a mess up? Is it something that went wrong with the experiment that one time? Is it an outlier? Why is it an outlier? Or maybe it's a very interesting thing in its own right that tells us something very important. It's up to us to figure that out, and we'll use methods like these to figure that out, principal component. So what I want you to do now is take a moment and go to our class website. Please, I'm going to pause to and go to our, if you haven't already done it, to our little web app. Okay, so the web app that I've put on our class website is you know, if we were in our face-to-face class, we would just go and talk about these things and draw them on the board and do all these things. That's a little clunky to do in this kind of format. So what we've got is a web app that's custom there for you to play with and twiddle with and move points around and see how the principal components respond. And we play with some data. So let me pop over to the web here for a second and pull that up, the web app. And it's important that you get this feel for it first before we go off and analyze, you know, huge data sets. We want to know why we're doing this. What's the purpose? So here I'm on week 7 on our class website, and the supporting material underneath the slides is the web app. I'm going to open that up, and this will open this little data set here. So you can read the text, of course, and it talks probably more coherently than I just did about what we're trying to do here. And it starts off with a simple 2D example where we don't really need principal component analysis, right? Because it's only 2 dimensions and we can plot it. Where principal component analysis comes into its own is when we have hundreds and hundreds of data points, right, and dimensions. But here, let's just see what it does in 2 dimensions. So we have some points here in this first plot, this x versus y. Now what we have in the second panel is the results of principal component analysis. If we apply principal component analysis on it, it's going to fit this vector. I'm showing it here in the first plot with the red PC1 and the green PC2. I apologize about the color code here. I should have picked different ones, but nevertheless, here's the first component. So it's the line that would lie closest to this data, and we can use that to describe the data. And we see that down here below these plots, this is how the data lies on the x and y axis, right? This kind of number line, if it was x and y alone. And here it is on PC1. See, it's most spread out on PC1, very little on PC2. PC1 is doing a great job of describing this data. But we can go and drag these points around, and we can see how the principal components change. You see that axis here in the middle of the first panel plot is moving as we drag these points around, right? So here's the idea being that now I've moved the points a little bit around, for example, and you can play with this and see how it is. This axis that lies closest to them now goes down a line like this, right? And we see them projected onto it here in principal component space, and we can see that, you know, our original number line, the points are not spread out, but look how spread they are here on PC1 and then PC2. So it's trying to do a much better job at displaying these. This new axis spreads the points more than the original ones. That's the kind of idea. So this is probably a little bit more motivating if we move to 3 dimensions here. So here I have a little rotating, because it's hard to show 3-dimensional data in 2D even on these websites. I've got this little rotating 3D plot. So there's clearly 3 cloud points here, but if I just plot them in any kind of x and y plane, you know, you wouldn't, if I plotted it like this, you wouldn't be able to see that structure in the data. What PCA analysis will do here, PCA will get these eigenvectors and it'll spread them out. So you notice where I'm looking is over here at the PC line versus the original x, y, and z. If I show the PCA results, this is the surfaces or the lines here that best describe the spread in the data. So if I have the points oriented like this, I'm going to see that spread and see potential structure in my data more clearly than I would in the x, y, and z. So notice here that now I can see for the first time this 3 kind of groupings or the pattern in our data, the structure in our data. that I can see on PC1 that I couldn't see it up above here on the original X, Y, and Z planes. I might have been able to see if I could. I can see if I zoom around and I'm able to do this. Of course, we can't do that in 1,000 dimensions, as we'll see in a little bit. But PCA is trying to show you that structure that's there. So have a play, see if you can get a feel for these things, see if you can find out how the PC results would look in these cases. And then what we'll do is we'll move on to a pretty small data set. This one, you know, pokes fun at the part of the world that I come from, in that it's goes and collects data about what people like to eat in Ireland and in the UK. So in England and different parts of the United Kingdom, Scotland and Wales and Northern Ireland, where I'm from, we eat pretty weird food sometimes, but you know, we think our food's better than their food and all the rest of it, right? And we think we drink better beers and all the rest of it, and we have better candy and what they call confectionery here, or soft drinks, is the sodas, things like that. So there's a little bit of different terminology, but we all think we eat slightly differently. But then we look at the data that's been collected here by this government agency in the UK, and it's like, it looks the same, right? People are eating the same stuff. You know, we look at these little plots, like the one above my head here, this bar plot, and it's hard to figure out, you know, what's the difference, right? Are these countries just all the same? They certainly look like it in terms of these 17 food categories. But we can take principal component analysis and do it, and what we'll see is PC1 does a pretty good job of describing this. And we see that Wales, England and Scotland actually are quite similar to each other, but Ireland, the north of Ireland, is much more different than those other three countries, and it's actually much more similar to other European countries if we would project them into this data, as we'll see in our second section of the lab sheet. So let's please go and do that. Let's take a couple of minutes to have a play with it. Here's the plot in 2 dimensions, where Ireland is over here, and these are these other 3 parts of the UK. I will give you a bit of time. You can pause the video, of course, and go and play with this little web app and have a look at it. When we get into our lab session proper here, so this is the hands-on lab section proper, we begin by actually going and analyzing that data. Okay, so feel free to pause me. That sounds like a good thing. Then we'll go and we'll start analysing this UK food data. Here I've got a little map of this part of the world, so you know what these countries are. Here's Ireland. This is where I was born, right on the border between north and south, right here in the north of Ireland. There's England, Wales and Scotland, all coloured up. And here's that same plot that we're showing. And the countries look pretty similar, right? They're eating quite a lot of meat. Their cereals, fresh fruits, quite big. Alcoholic drinks, of course, is quite big. And you know, what's the deal here, right? Are some of these countries? What's the pattern in this data? It's hard to tell, even in this teeny tiny data set. It's only 17 dimensions. Remember, we have hundreds and thousands of dimensions in our data set. If we can't analyze 17 dimensions, what's the hope for us, right? So I'm going to pop over to our studio, and we're going to start doing some analysis of this data. So I'm just writing a title for myself. The single pound symbol here, a hash symbol, will make a level 1 heading, so big text heading for this section, principal component analysis. First we will look at Okay, so we're going to look at this UK food. PC of UK food data. I'll maybe give this a level 2 heading. First we'll look at the let's not call them weird. Food folks in the UK eat. Okay, so first thing I need to do is get the data in, and I provided a tiny URL link on the class website, the lab sheet to that. So that URL link, I'm going to just copy it from the class website here. So here it is, tinyurl.com, and then capitals UK dash, well, you don't actually need the capitals, UK dash foods is the link, and I can read directly from that web page. I should call it the same thing I did before. I'll just call it X. Here it's a CSV file, so I'm going to use read.csv, and I'm going to give it URL as the input, and then I'll just have a little look at the head of X, so the first few lines of X here. So I go and read it, and I see that I've got some columns. I've got a first column called X, and I've got a column called England, Wales, Scotland, and then last but not least, and Ireland for Northern Ireland here. And then there's some values down the rows for different foodstuffs, cheese and fish and whatnot. Now the first thing I notice here is that this first column, this is really what I want to be row names. If I looked at X, if I clicked over here in my environment panel over in above my head in the top of the window up there and had a look at it, X, I would see that it's got row names 1, 2, 3, 4, 5, 6, 7, which I don't want. I actually want these categories of food to be the row names, like cheese and meats and fish. I want those to be the row names. So what I'm going to do is actually I'm going to change this code here. I'm going to actually say row.names equals 1. Please make the first column, number 1 column, be the row names. Okay, so when I run that again, let's make our window big so we can see it. When I run that again, I should see here that now the X has disappeared above, and I have those categories as my row names. We can check X, which I've already got open here, and see that it's now changed. You see the way these row names are now in the grey here, and they're not as separate columns. This looks great. This looks good. I've got my data in, and we could explore it. We could plot it in lots of different ways and see if we can find out differences in this data set. It's only a small data set. Only 4 columns here, 4 different countries and 17 different rows here. So it's not a huge data set. We should be able to find some patterns in this data set, but really it's kind of difficult. If I go and try and plot it up in lots of different ways. For example, if I do a bar plot of X, right, and I'll have to make it a matrix for this. Let's do it as matrix X, and I go and do this. So that's a ugly stacked bar plot, right? Which is helpful to no one, but let's maybe make the colors. What I'll do is I'll color it up, make it at least try and cheer myself up a little bit. Colors. Let's pick colors from the rainbow palette. Rainbow, and we'll do the number and row of X. So give me 17 in this case, because that's the number of rows. 17 colors from red, green, orange, yellow, the colors of the rainbow. Still a not very helpful plot, but here each color and each category in the bar plot is there's 17 of them for those 17 foodstuffs. We can see that maybe just Wales eats more, generally consumes more than any of the other countries here in this region, but really it's not particularly helpful because you can't see, these lines all start at different points. So one thing we can do here is we can argue with this plot. Let me take it again, and we'll do here we'll argue with it and say, rather than being stacked, let's do beside equals true. And if I do this one, I get a plot where I have England, Wales, Scotland, and Northern Ireland. And I can see that the categories that are big in England are the same sort of categories that are big in Wales, or the same sort of categories that are big in Scotland and Northern Ireland. There's some difference, right? This green one here is slightly bigger in Northern Ireland. This purple one's bigger in Scotland. The blue one's bigger in England. But it's pretty hard to see patterns here, even in this teeny data set. One plot that is kind of useful, it turns out, when you have small data sets, which we don't often have, is a so-called pairs plot. So this is our pairs plot, and we can give it here our input x. Oh, if I type in the right place, excuse me, x, and I'll give it the same colors as before, so that rainbow palette, the n row of x. This would be our and I'm going to try that one out. And maybe, so here it's got the default plotting character, which is the unfilled circle or the open circle. I'm going to change that PCH to be the filled circle, 16 in this case, to be the filled circle, or 15 the square. And what we've got here is a bit of a, if you've never seen these plots before, they take a little bit of explaining, but what we've got is, of course, down the diagonal, we've got the names of these countries, England, Wales, Scotland, Northern Ireland here. And it's every possible plot of England versus Wales. So this would be this first panel, if you can see where my mouse is here, would be the plot of England along that up-down axis, along that y-axis, and then on the x-axis would be Wales. So it would be the plot of every food of the 17 foodstuffs, the value in England versus the value in Wales. And if they were exactly the same, if people in Wales had exactly the same amount of these different 17 categories of foods as people in England, all these points would have the same value, so they would lie along the diagonal, they would lie along a straight line in both of those. And now where you go to this plot, for example, and if we go across that first row and the last column, what this is England again, right? So we're reading across the first row, so those are all plots of England. And this last column along the bottom is Northern Ireland. So this is all Northern Ireland here against these different countries. So this is a plot of England versus Northern Ireland. And we see some points that are clearly not along the diagonal. This blue one and this green one, for example. What this means is, if it's above the diagonal, there's more of it being consumed in England. So there's more of this blue food category. We don't know what it is yet, but we can find out in a second, being consumed in England, and there's more of this green one, pretty fittingly, being consumed in Northern Ireland here, right? So the Irish are eating or drinking usually more of whatever this is, and the English are eating or drinking more of whatever this is in the blue here. So we could start to learn something about the data. We can see that they're mostly along the diagonal in this first panel, mostly along the diagonal here, with some scatter, but really the Northern Ireland does look kind of different than the other countries. We don't really know why yet, or if it's consistent across them, but we're starting to make inroads. But we had to do a lot of work to get to a plot like this and figure that out. And typically whenever we're dealing with really large data sets, much larger than this, it's not practical to do this kind of plot, right? If we've got 20,000 odd genes and hundreds of samples, it's not going to be a very useful plot to look at. Okay, so enter PCA, kind of PCA to the rescue here. So let's start doing our principal component analysis of this data set. So we're going to go and run the main function for PCA called prcomp. Here in BSR, there's lots of principal component analysis functions in add-on packages, some of them really nice that do it, for example, for genomics data and bioconductor, or biomolecular data and bio3D and other packages. We're going to just keep the one that's always there for us to use, and if we can use this one, we'll pretty much find the other ones easier to use because they're a little bit more straightforward than this one. But this one comes out of the box, it's pretty efficient, and it works well. The only issue, let me write the notes. to use the main PCA base R function, which is called PRComp. We need to take the transpose of our data. You know, we often will we'll take our data and just like we have it here, or you know, as we'll see with our RNA-seq data in a little bit, you know, we have our genes in the rows. Here are the foodstuffs in the rows, right? The things that we're actually measuring, then the conditions, like the drug treated or the wild type and the knockout down the columns. But this base R function actually expects it the other way around. It wants the rotation of that, the transpose of that. It wants things to be in a different order. So we're just going to have to play along with it. You know, we read the help page and we figure out that that's what it wants as input. So we're going to take I'm going to call I'll call the results PCA. We're going to take PRComp here, and here I'm going to have I can't just give it X as input. I have to give it the transpose of X. So you can play with the little transpose function here to see what it does with matrices. And then I'm also going to give it scale equals true. We'll talk about scaling and what it does. This is, if you remember our rescale function from when we were writing functions, this is going to do that for us. It's not going to, if things were measured on completely different units, they would, and I need to spell it correctly, this will take care of it. It'll scale it to be mean centered and unit variance here. So if I do this, I can do, and I'll print a summary of the PCA results, my object here. So I'm going to run PCA. Oh, again, typos, everyone does them. So I've run it, it's done this PCA analysis for me, and it's printed out this summary table. So let's have a look at this table for a second. Actually, let's look at it now. So it's telling us here that PC1, PC2, PC3, PC4, it's got here. What we've got here, where we often look, is this second line and the third line on these outputs. This tells us the proportion of variance. Remember, what the PCs are trying to do is these lines that are these new axes that maximally describe the variance in our original data set. So this is the variance of the original data set that are captured along these new PCs, or these new eigenvectors. And it's done a pretty damn good job, nearly 70%. So you multiply these by 100 to get percent values here. There are 0 to 1s. Nearly 70%, 68.33% is captured in the first dimension, and then almost 25%, 24.8% in the second dimension. The line below, the cumulative proportion, that is if you add up these two numbers here, if you add up the PCs up to this point, the cumulative values, so that's 0.6833 plus 0.2487, you get over 93% of the total variance is captured in these first two principal components. So that's pretty awesome, right? We've taken this data set and we've got pretty much everything that's going on in 2 dimensions, actually in 1 dimension, 70% odd in 1 dimension. So we can go and make a figure now that's going to be a better display of the results than if we plotted all the original things, right? That pairs plot or any of the other kind of figures that we could do. Let's go and do that. So we're going to, let me turn this off for a second so you can see the code. And we're going to make our main PCA result plot. People call this a score plot or just PCA plot. Scores, they're the scores along the PCs, along these new vectors, or the projection of the original variables onto these new PCs. There's a lot of terminology in this that describes, it sounds complicated, but it's really just describing a plot of PC1 versus PC2, right? So to get to these things, we need to figure out what's in this result. We've called it PCA. I'm going to do attributes of PCA like we did before, with the result of Kmeans, for example, to see what was in it. We could look at the help page too to figure out what these are. There's a thing called standard deviation. Of course, we could square this to get the variance. That's something we would need to make our scree plot, for example. And then you've got a few other things. Scale is going to be, true or false. We said scale, please. True. And this thing x, this is what we're actually going for in this case. So we're going to use x to make our PC plot. So I'm going to do, well, first off, let's just do a summary plot of PC results. How good are we doing? Results. So if we just do plot that PCA result object, that's going to do our scree plot for us. So here's with our PC1 being the most, PC2, then PC3, and then little else. So that's not the most exciting thing in the world, but that's our scree plot, or our loadings plot. Here I'm going to do the $x component, PCA $x. This is what we want to plot. So I'm going to do plot of PCA $x, and I want the first column of this. This will be PC1, and I want to plot it against the second column of this, so x versus y. And I'm going to do the second column here. So when I plot this, it's going to be my PC1 versus PC2 plot. 1, 2, 3, 4. There's our 4 countries. Okay. Now, these aren't, I need to know what countries these things are, right? So I could do that. I could do my little plot here of, actually what I'll do is just add text to it. So if I do text and I use the same XYZ coordinates here, so I'm just going to copy this. So this is going to add text at these coordinates. And then the labels that I want to actually add are going to be the call names, because that's the country's names. So call names of X, if you remember X, if I open it up here, it had England, Wales, Scotland, Northern Ireland as the column name. So this will put those names on the plot, and I can see it's a little bit clipped here, but here's Northern Ireland off by itself, then Scotland, England, Wales. And remember PC1, this first axis was the one where most of the action was, and it's showing us that Scotland is closer to Northern Ireland than England, Wales, but this really is quite separate from the others along that PC1, which is the most important axis. We always remember that this is describing more of the variance than the second principal component. That's separating Scotland, for example, from England and Wales here. So we could go and color these up. Let's do that. Perhaps let's make a little color vector, and we'll color them by let's call it my calls or country calls. We'll color them by the kind of colors they play in their rugby jerseys. So England's first, they play in white, which is not going to be very useful, so I'm just going to color them orange, put them in a change strip. And then the next country here was Wales. They play in red, and blue for Scotland, and Ireland, of course, in green. So then we have red, blue, and green. So I'm picking these colors. I can do then color equals this thing that I've just made, this country calls vector. And I can actually use the same line here. This will have the effect of making this plot, and I could increase the axis, the X limb here, for example, a little larger. Let me put this over multiple lines so we can see it. So here's the column. Actually, we don't want to do it there. We want to do it in the original plot. So here I'll do X limb equals, and I'm going to increase it to -5.5 or something like that. minus 5, 5. So if I do that, that will increase that axis so we can see the labels. There's Northern Ireland, Scotland, England, and Wales spread out. Okay, so you might notice that these results actually are looking good. We scale this data, here. Let's take this off for a second. Scale, and let's run these again. Here's our 67, 29, so 96% of our variance. Here's our PCA plot, same as kind of before. We can make our figure. Let's take the XLim out here for a second. And we get our plot. So notice now something's happened, right? The scale axis, we haven't rescaled them. You see the values, they're not 0 to 1 scaling, and they're not mean centered. We get this plot like the one on our website, because I didn't use the scale function on our class website. And we see here, just as the values lie, Wales, England, Scotland are over here, and Northern Ireland's off, and it's Todd over here, different. And I'll add some axis labels on this. And also the order of these things split. If you do this on your computer, you might see that Wales, England, and Scotland are over on the plus side, and Northern Ireland's on the minus side. The sign of these things are arbitrary, as we'll see when we talk about how this method works in a moment or two. Okay, so the sign's arbitrary. Scaling, what effect does that have? Let's have a look here at the slides for a second to talk about scaling. This is a kind of important side note here to the principal component analysis method. So here is an example where clearly scaling is required. If we take, remember our automobiles, our cars, our MT cars data set. So there we had things like the miles per gallon, the city miles per gallon, the horsepower, the number of cylinders in the engine, the displacement, these sorts of things. They're all measured on different units, like the number of gears, the transmission size of the car. It's measured in units that are very different than the number of liters of the size of the engine, for example. These are in hundreds, and horsepower is in hundreds, and cylinders are in single digits, right? Usually. So these are definitely going to require scaling here. If we didn't do PCA with scaling on this, it's going to look different than it does look with scaling quite dramatically. Here in the line of code on the below where I print out the head of MT cars, I'm looking at the means and the standard deviations of the columns, and I see that they're different. So when the means are different and the standard deviations are different, when they vary a lot, like they do in this case, we're going to have to scale the data. Otherwise, your results are going to be dominated by the original variable that has the most spread, right? Because that's what PCA is going to go and look for, the spread in the data. And if the biggest range is horsepower, it's going to be dominated by horsepower. You'll go see your results be dominated by that variable when we get our PCs. So here's an example. If I do PR comp on the MT cars without scaling, everything's squished in, and the cars that are the most largest horsepower, the Ferraris and the Maseratis and things like that, are going to be the most spread out from all the others that are squished in here, like the Hondas and the Toyotas and things like that. If I do scaling, if I make it more equal and more fair, if that's what you're looking for in this case, then we get a much more informative plot, like here. Okay, we see now interesting patterns where we're at the Honda Civics and the Toyota Corollas, and the small size engines are down in certain region together versus the big kind of beasts over in another region of the plot. And the same sort of thing that we saw in our PCA of food data, right? So let's go and build up our complexity a little bit and go on to another example. Okay. So here I'm going to go back for a second to our class website and see what the next section was. And I want you, of course, to go through this website and answer the questions as you can to make these figures. And you'll see how to make we're digging into the loadings, which is another important thing that's often overlooked in PCA. We can actually find out how the original variables contribute to these new PCs. That's important because it'll tell you how these countries actually differ or why they differ, right? So here you can see fresh fruit, alcoholic drinks. Those things discriminate countries of the UK, whether you drink a lot and eat a lot of fruit versus whether you eat a lot of potatoes, right? So you can see some patterns start to emerge and some stereotypes be reinforced here, for example. And then there's these biplots that often aren't useful when we go to larger data sets. So let's go and do one more example before we talk about how PCA really works. We're going to do PCA of RNA-seq data, this little example data set. So here we have some data that we've got 5 wild type experiments and 5 knockout, where we knock out a gene in this data set. We do our RNA-seq, our sequencing. That's actually going to be the topic of our next section of the course, how we get data sets like this and how we go through from raw reads from a sequencer to analysis. But for now, we're just going to take the count data. I'm making it available at this second URL here. We're going to read it in, we're going to set the row names, we're going to have a look at it, and then we're going to go and do PCA of this data. So I'm going to take this code, which I provide, and we're going to go back over to RStudio here and start to analyze this data with PCA and see what we can get. Okay, so slightly more dimensions here, of course, slightly more interesting data. So here it's PCA of RNA-seq data set. So we're going to get the data from online, and then we're going to start processing it. So I'm going to paste that in. Here's the URL. It's at this tiny URL expression -CSV here. It's also on our class website, linked to if you want to download it and put it in your project and read it that way. I'm going to check that this works. It goes green. It shows me a summary of it. I see that there's quite a few columns, quite a few rows. I'm going to check how many there are. So I'm going to say how many. Let's give myself some room. So how many genes? I've split this into a much smaller data set on purpose. How many genes do we have, and how many conditions? Like wild type, knockout, these things. To do this, I'm going to look at, you could look at nrow and mcol separately. I'm just going to look at the dim for dimensions. And we called our data set here RNA data. So I'm going to look at the dim of RNA data. And that tells me it's 100 genes and 10 conditions, 5 knockout, 5 wild type, 5 knockout, only 100 genes that we've limited ourselves to in this case. So let's go and do a PCA of this data set, and we'll use our scaling here. Okay, so we're going to do PCA of this data. We're going to use the prcomp function. So let's call our result, what did I call? Well, just call it PCA again, overwrite the previous one. So this is going to be PCA here of prcomp. We're going to take the transpose of our data set, the RNA data. I'm going to say scale equals true, and then let's get a little summary of how well we're doing. So when I do this, I see, and let me show you the code to read the data in case that went by too fast. It's read CSV, the URL that we've got. I'm setting the row names so the gene names actually become the row names rather than a separate column. And then I do my PCA, and I see that, whoa, over 90% of the variance, the original variance in the data set, is captured in one dimension, and only 2% in that, 1% and less. You see they're dropping off, like falling off a cliff here, right? So the first PCA is really where all the action is. If we did a little summary plot of this, we'd see it too. So 90% to almost over 92% is captured in the first dimension, PC1 plus PC2 is almost 95%, 94.9% here of what's going on in the original data. So if I just do plot of PCA, the scree plot's going to be dominated by that first bar, that PC1. We can see that here. So let's go and make our figure, our main PCA figure, our main PCA result figure here. And to do that, we're going to take our plot function just as before, and PCA is our result object, and it's the $x component that we're going to look at. The first column is PC1, second column is PC2, third column is PC3, fourth column is PC4. You get the idea. So we're going to plot first versus second again, and see where the data lie. Okay, so here when I do this, there's points clearly separated from each other. There's points over here on the minus side, there's points over here on the plus side along this first principal component. And if I count them up, there's 1, 2, 3, 4, 5 points in each group. 5 wild type, 5 knockout. This is something that we'll do quite regularly at this stage in like an RNA-seq analysis or other form of analysis. Plot the data. Step 1 is always visualize the data. And PCA is helping us here. Rather than viewing all the genes and all the samples in some crazy way, quick PCA analysis, as we've just done, and look at the results. Are the wild type consistently different from the knockdown? If they are, the knockout, or are the drug treated and the controls clearly different in this kind of analysis? If they are, happy days. We're good to continue, right, and dig into why they're different and go into the biology of these things. But if they're not, or if there's some outlier or something that's causing a problem or driving all the variance in this data set, then we probably need to pay attention to that first. Okay, but here let's make sure that this is the case. Let's color them by whether they're so the first 5 here samples, when we looked at it, they were the wild type ones. So the first 5 were wild type, and then the second 5 were knockdown. So we could make a little color vector of 5 colors, and then another vector of 5 colors. We could do that. That would, so let's say my calls, and let's say repeat red 5 times. And then we'll also add, or we'll do what colors will we do them? Black and red. Maybe we do rep black 5 times. So the wild type, that was first, right? The wild type, when I look up here, we could, I think on the workbook, on the web page, I pulled the colors from the actual names with a little bit of code, but we'll do it more simply. So it's going to be 5 blacks colors and then 5 red colors. Here, I'll just do it that way. So this is our little vector. Let's print it out just to make sure you see what's happening here. So my calls here is going to be black, 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 red, 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 and it's going to be length 10. So when I go and use that to color up my plot, call equals my calls, and let's make the points different. PCH 16, that's filled. Plotting character. So when I do that, I see that here's my knockdown. Sorry, here's my wild type, and here's my knockout experiment. So they're clearly distinct, and indeed 90% is captured in that PC1. If we did right, xlab equals PC1, ylab equals PC2. We label them up. We could put a title on it. We'll do all those sorts of things. So we've got a pretty striking result here, right? This is clearly showing us that the knockdown is consistently different than the wild type in terms of these eigenvectors, these better axes that describe our data in a better way than all those original variables. We could dig into it just like we did with the UK food data in the workbook and find out, which foods are distinguishing these countries along these different components, like which foodstuffs, the alcohol and the potatoes and the vegetables that make Ireland different from the rest of the countries. We could dig into this and see which genes are making the knockdown more different than the wild type, right? Which are the other genes that are contributing to this. We could look at the loadings for those original variables, which in this case are the genes, right? Our observations in this case. So let's go and have a look to see if we have any questions here. There's some code in the workbook in the lab sheet where we can do these. Let me pop over here to the website. We'll see that there's code to do this with. So here's the kind of plot that we've made. We put some labels on it with this text command. Here's the call names. Again, we're using as before. Position, we're saying just make it to the left or make it to the right. If you're going to make nice figures of these things, not the kind of exploratory, quick, dirty plots that we're making at the minute with base hour plots, we could go and use ggplot. I've shown you the code to do this here. Of course, you need a data frame to use ggplot. And so here's the kind of idea. We build up our plot step by step and can make pretty nice looking figures, things that you would see in manuscripts and publications and the like this kind of figure, following that same recipe that we covered back in that section of the course, but using a data frame here as input. So let me pause here before I go too deep into this and lose more people, and see if we have more questions. So Lee asks, what are the values inside the X of the PCA result? What they are, Lee, are these are the scores of the original observations on the new PC axis. So each column is going to be in $X is going to be PC. So it's basically it's the results that we're looking at, right? So it's the new, it's the view of our data along our new axis is what it is. Okay, so it's the scores of those original observations on the new PCs. So we would plot to get PC1. We'd access the first column. So we're using PCA $X square parenthesis blank because we want all rows, comma, then 1. That's going to access the first column. And then we're using number 2 to access the second column. We plot 1 versus 2. We'll see that PC plot. Okay, if you wanted to plot PC1 versus PC3, right, we could do that too, but we'd use the number 3 there to access the third column of that data set. Okay, so with that, I'm going to pop back over here and we'll see. We can keep working on this. The step here, let's do this before I forget, because I will forget if I don't do it. We're going to go figure out how PCA does this, what this $X is actually and where it comes from. Really answer Lee's question in more detail here in a second. But I want to remind folks that the third step here, I haven't actually answered many of the questions for you. I went through it at a breakneck speed here. But the third step is producing a PDF report of our work. So what that means is that we go back to RStudio, of course, and we have our data here with all our narrative text. I save it, so you can click the little save icon or Command S or Control S, and then we knit it. And at the minute, because I set it up this way from the start, it's going to make a HTML report. Let's see if it works, or if I have error messages here, I need to go fix them. So here's my report. It's got my my text and my code, and it's got some figures in it. And our PC results. I want to change this from a HTML to PDF, and you can do that in a couple of ways. You could just click the little knit drop down optional little extra arrow here, or whatever you want to call it, beside the knit button, then you can knit to different formats. What I'm going to do is actually do it properly, or the old school way. I'm going to change back up here right at the top of my document. I'm going to change where it says HTML document just to PDF document. I'm going to make sure my name is in here at the start. I'm going to add with my name my UCSD ID number. I don't actually have one, but please add yours. So my name plus my number. This just makes sure you get the credit for this thing, and because if we can auto pull the UCSD ID in case your name's spelt wrong or anything. So if you have 2 data points, it's easier to make sure everyone gets their due credit. And I'm going to save that. And it's got the key change I made, of course, here was just PDF rather than HTML. And I knit it, and it's going to go off and make a PDF report rather than the HTML report in this case, assuming that you have knitter and tiny text installed correctly. If you are struggling making PDF report, let me show. So the PDF is just popped up here. So here's the PDF. You can see with much cleaner document that you would want to share, with your boss or professionally. Although the web page is good too, I often work with the HTML reports, as you see. But this looks pretty professional, much more so, and it's actually the only format that Gradescope will accept, which is why we're doing it in this way. So it's got my code that I can see. It's got my comments in it. Hopefully my answers to the questions, and I can see that we're working through these things and making progress. I'm going to go back to the website for a moment. And so the idea here is that you make that PDF report and then upload to Gradescope. We'll open that up after the class. The Gradescope submission will be available for you to put your PDF report on there to get feedback from the IAs and myself. Okay. Also note, before we dig into PCA a little bit more, there is, because I realize that I'm kind of spoon feeding you this a little bit in this format, there is a mini project that we have on some cool data, some cancer cell data. Let's go and have a look at that for a second before I forget about it. So on our schedule, here are homework. This is one of our last data camp assignments that we have, Introduction to the Tidyverse. We have extra time to do that. But here, this mini project, if you click here, this will bring you to a data set and discussion of a data set that we're going to analyze. This actually comes from biopsies from what's called FNA, or fine needle aspirations of breast masses, from patients at the University of Wisconsin Medical Center. And those cell nuclei that are visualized there, they've been encoded in various ways. And our task is, much like the pathologist, to figure out, is there something different if this turns out to be benign or malignant? Can we use these methods that we've discussed, the clustering methods and the multidimensional analysis methods, these principal component analysis in particular, to make sense of this data and actually help in this task? So again, this is unsupervised learning that we're doing. We actually have the labels from the pathologist, the expert diagnosis of these patients, and we'll use that to kind of bootstrap ourselves to help to see how well we're doing. We walk through, and you have to complete this and make a separate R markdown document, separate report. You have a couple of weeks to do this, so it's not all today. But we go through, we read this, you download this CSV file into your project, your new project, read it in, set it up properly with the row names. We'll see that we have malignant and benign samples in here, quite a few actually, people's data, souls are in here. And we do our PCA analysis result and we color it here by diagnosis. We see something pretty remarkable, right? Let's actually scroll down to our ggplot. So here we see along PC1, which is a great job of describing this data, we see a separation of the red here, which is for benign, and the cyan or green-blue color here for malignant, the cancer samples. So this is really amazing, right? This is kind of striking, just like it was in those other examples too. But here I think it's much more clear to see the significance of this work, right? The importance of this. This is actually showing us some structure in the data that's really hard to pull out in any other way. You can plot and dice and slice and make figures to the cows come home of the original microscopy data, right? The radius of the nuclei, the roughness, the whatever other things that these pathologists are looking at and see them. But it's not until you put all those variables together and do this kind of principal component analysis that you really see this clear pattern that we could kind of draw a line down here, right? We could draw a grouping, or what we'll do is we'll cluster in this space, and we could cluster and see benign and malignant samples here. This could be really helpful in the analysis of this sort of data. And of course, we're using it here for illustrative and educational purposes. But the idea here is that we'll go and we'll dig into this and we'll start to see how useful PCA is and make these kind of figures. We'll walk you through this, and you'll get to struggle through this as well and learn about the importance of this technique, all the while thinking about, is this helpful for the analysis of this type of data, and how could you then take these methods and apply them to your research questions, right? If you're in the lab or you're working in some company and you've got some complicated new data set that's coming in, could you take these methods and start to reveal otherwise hidden structure that using these kind of approaches? They're very, very useful, very, very commonly applied. It's really important at this stage, not that we understand the math necessarily, that we'll talk about now in a second, but really understand the purpose of these things. What are they trying to get at? And we can, of course, always build up our knowledge of the behind the scenes as we go through. So please do explore that and have a go at it. We're actually combined methods. We cluster on the results of PCA, and we look at sensitivity and specificity for detection of cancerous, the malignant versus benign. So it's a really nice data set to get your teeth into, hopefully, and motivate you to learn about these methods. Okay, and of course, use Piazza, use the IAs, and myself to help you delve deeper and get a deeper understanding of this kind of topic. More so than the very guided, spoon-fed thing that we're doing in these lab sessions. The project will really help you take these methods and apply them in a much more realistic project-like setting. Okay, so to finish off the last handful of minutes, how the hell is this working? Right? How are we able to get these kind of beautiful results? What's happening here, right? Each dot here, I should have described to you, each point here in this project represents a soul, represents a person, right? Represents their biopsy data. So each point is one of these rows in our data set. And what's shown here is all of the data together, and it's all summarized in one point. How did it do it? In our thing that we walked through together when we did our RNA-seq analysis, each point ended up being one of those samples, the knockdown, the knockout, or the wild type, right? With all those genes in them, all being summarized to one point. Or if we did our UK foods, each point was a country, right? It was all those foodstuffs, of course, but we've summarized it down to one point. How did PCA do this? How did it compress this data essentially and make a useful display out of it? Like this one, where we can start to see for the first time that there is something distinct about these cancer biopsies versus the benign biopsies, right? How does it do that, and what can we learn from it? So let's go to the slides for a minute, and we'll talk about this for the last section of our screencast. So these slides, I've actually, we would talk about this and work it out on the board if we're in a hands-on session. What I've done is I've tried to adapt it to this slide format, and I'm borrowing heavily from Josh Stammer's excellent description of this. You can find it on YouTube as well. So I'm going to kind of follow in his mode of describing it step by step. and see how we get on. So Josh, when he talks about this, he shows this single cell RNA-seq data set. What's shown here is it's a graph of RNA-seq data. It just happens to be single cell data in this case from quite a few years ago. But there's 10,000 odd genes in this data set, so that would be the rows in the original data set. And there's counts in the different columns, different things that they're observing here, that they're actually studying. But what's happening is each dot in this plot that's in their paper represents a single cell on its transcription profile. And the general idea, just like with us, if our points from our cancer patients are all together, they're similar in their cell type, just like it was in our RNA-seq data with our knockdown or wild type. If they're close together, they're more similar to each other than they are to the other ones. It's the same deal here. So each dot represents a transcription profile, and the general idea here is that similar things are going on with their genes. Their points are going to be together. They're going to cluster together, and they've drawn these kind of circles around them. These are blood cells, these are neuronal cells, these are epidermal cells, etc., because they cluster together in this PC space. And it's really an amazing result, right? This insight that it's giving you from these huge 10,000 gene data sets, right? So PCA, what it's doing, it's compressing this data, a lot of data, into something that captures this essential essence and tells us something that we want to know about our original data. So PCA, let's recap, it takes a data set with lots of dimensions, i.e. lots of cells, and flattens it down into something we can plot, like a 2 or 3 dimensional kind of plot that we can look at. It tries to find meaningful ways to flatten that data, to compress it down, by focusing on things that are different between cells. That is the variance, right? Remember, it's trying to capture these axes of maximal variance. So if they change the most, if there's the most variance in them, that's what PCA is going to go after, right? So here's an example of what that data might look like. So we've got our genes down the rows here, and we've got cell 1, cell 2, cell 3, cell 3, however many cells that we've got, and we'll have 10,000 genes, for example, down the rows. So there's numbers in them that describe, for now we'll just think of them as counts, how much of that transcript was there when this experiment was done, okay? So if it was just 2 cells that we were looking at, 2 columns, we could just plot it like we've done before, right? X versus Y, cell 1 versus cell 2, and we might see something like this. And here, generally speaking, the dots are spread along a diagonal line. They're actually showing some correlation here, some positive correlation. And another way to think about this is that the maximum variation in this data is between the end points, the 2 tips of this arrow that we've drawn here on this slide. And there's also some spread above and below, right, that we could capture with another vector, another axis here, what we'll call PC2 in a moment, for example. Another way to think of this is that the second largest amount of variation in this data set is captured and is signified by the ends of these 2 arrows, or of this arrow, the second arrow that we've drawn. Now we can draw a new graph here using these 2 new axes and get rid of those old axes. So in general, it varies a lot left to right, and it varies a little up and down, and we're capturing that here with these new axes. The first axis, the first new line here, which we'll call PC1, the data varies a lot from left to right, and in general it varies also, but less in this up-down direction. Note, importantly, that all the original data can be drawn in terms of these 2 new lines, this up-down or left-right variation in this 2D graph. That is to say, we don't need the original lines or any other line or diagonal line or anything else to draw this data. We've already captured it all with these 2 new axes. We call these axes, of course, PCs. PC1 is the most important, PC2 is the second most important axis of these things. PC1, PC2. So for general idea so far, for each gene we plotted a point based on how many reads there were for each cell. PC1 captures the direction where there's the most variance. PC2 captures the direction with the second most variation in the data. So for now, let's just focus on the first one, PC1. We'll come back to PC2 in a moment. So, as we said, the ends of these data points capture the biggest spread, right? It's determined the kind of, if you were going to draw this line, really where you put the tips or where you orient the line is really determined by these points at the end, right? These circled genes have the biggest influence on this line, its length, and indeed its orientation here, right? So the length and direction of PC1 is mostly determined by these circled genes. We can score any of these original red points, these genes, by how much they influence this PC1. So if I label some of these genes up, we'll call them A, B, C, D, E, F, etc. We can say A in that circle and F in the circle on the far side, or at the lower circle, they have a high influence on this vector, this PC1. Whereas those genes in the middle, B, C, and D, have a low influence, right? They're not close to the ends of this line. So some genes have more influence on PC1 than others. And we can actually put this in numbers. We can say the center of that axis is 0, and then we can move along in the plus direction or the minus direction. So from the center of that little vector that we've drawn, we'll have 0. And then we move in the plus direction, we're going to have high values. So A is going to be plus 10, whereas F is also high, but it's going to be in the minus direction, like minus 14, as we're saying here. Okay, so that's the idea. These extreme genes on the ends of the line give the large either positive if they're on one end, or negative if they're on the other end. And actually, you know, which one you say is positive and negative is kind of arbitrary, as long as you do it. That's the important thing. And we would do the exact same thing for the other vectors. So here I'm doing the same thing for PC2. So in this case, B and C have a high positive influence, and D here, sorry if I go back, has a large influence, but in the negative direction. It's getting a minus 12 in this case in our little table here. So what have we got so far? We've got our two PCs, and we've got our influence in numbers on these vectors. Okay, so what we're going to do is we're going to go back to the original data, the count data for all those genes, and this influence tables that we've just made, and put them together, combine them to get our results of principal component analysis. So here's our original read data back in this first table. And what we're going to go through here now is we're going to take cell 1 and we're going to look at PC1. So we're going to take cell 1 and its read count, so a value of 10, and its influence in 10. And we're going to multiply those together. So it's going to be 10 times 10. And then we're going to go on to the next gene, B, for example, and do that. So here's 10 times 10. That's our counts by the influence on the PC here, and we're going to do the same for the second gene B, so that's 0 times 0.5 here, and we would do the same for the next gene C, that would be 14 times 0.2, plus for gene D that would be 33 times 0. I'm reading off from the two tables, and I get some answer, for example 12 in this case, when I sum all these products up. And that's the score on PC1 for that column of data, for cell 1, that original column of data. And I would do the exact same thing for PC2. I would go on 10, and now I go to my second table for PC2, 10 times 3 plus 0 times 10, etc., all the way down for all the genes here, and I'll come up with a value. In this case, I get a value of 6, let's say. And I can take that 12, 6, and I can draw cell 1 on our main result, our PC score plot, our scores of these things on the PCs. So here it is, it scored 12 on PC1 and 6 on PC2, so I use that as my X and my Y axis, and I've got a point for cell 1. And I can do the same thing now for the second column of original data. I could do the same thing, and I would get some values, and for example, if I got 2 and 8, that's where it would lie. If I did if we sequence a third cell, third kind of condition here, we did the same thing, we would get some values. And if it was similar to cell 1, it's going to lie in a similar location on this PC plane. It's going to get similar scores on these PCs when we take those products, the sums of all those products down. So hooray, we've got it, right? Cell 1 and cell 2 are similar to each other. And that is just the same thing that's happening here with that original data that Josh talks about in RNA-seq data set, where we now know that these points that lie close to each other, they have similarities in that transcription profile and where they're lying on these new PCs, these scores. And that's the same thing that we see with that patient data, that cancer data that you're going to see in your project data. So it'll tell you this similarity, this structure in your data set. Okay, so what I want to do is for you to, this is not all going to make sense right away, but we're going to use this project to start guiding ourselves into thinking more deeply about these topics. These are complicated topics that aren't often taught in these types of courses, but they are extremely useful for analyzing data. We're going to use them in RNA-seq in next week's class, in our next section, to analyze some real data that we're going to go and take all the way through from raw reads from a sequencer right the way to this kind of analysis. And we want to understand them. We want to at least know the purpose of these tasks to start to see structure in our data and understand how they work, okay, by doing these kind of projects together. So please give it your attention. I know it's a busy time for everybody, but it is important technique. It's a really useful technique that will come back again and again and again for all different sorts of data sets, whether it's protein structure, drug discovery, drug design, various types of genomics. We're always looking at big data sets and we want to try and understand them. And this is a great tool to have in our arsenal to do that. Okay, so with that, I noticed that we're approaching our, we're scheduled for 3 hours, but I like to keep this a little shorter, around the 2 hour mark. So I know we're approaching time, and we'll finish there for now. And thank you very much for your attention. I'm going to check one last time on Piazza. Of course, we'll keep the discussion going there. And actually, what we'll do, because it's 5 past, we'll just start to end this screencast. And yeah, thanks very much. We'll see you in our next set of videos. And of course, keep the discussion going on Piazza. And thanks for your attention. Bye now.